Today we are going to talk about chromosomes. And so what is a chromosome? A chromosome is a tightly packaged sequence of DNA. And so DNA, you could imagine if you were to pull, it would unwind from the chromosome. A chromosome is a very dense structure of DNA. And so if we look at a chromosome, there are segments of it where if we were to unwind that, a gene would be found. So we could call that gene A or B or whatever. This shape of chromosome is different than this shape, but they are still two chromosomes. So this is two chromosomes, and this is also two chromosomes. The way that you count chromosomes is by the centromeres here. You can see there's two here, and there's two here. You always have two sets of chromosomes. After S phase of interphase, you have this shape. And the reason for that is so that during mitosis or meiosis, you can split these sisters. So these are called sisters. And they're actually sister chromatids. Chromatids have the same gene and same allele, whereas a pair of chromosomes, so these together would be called a homologue. Homologues have the same genes, but different versions of those genes. And that's because this one would come from mom, this one comes from dad. And again, you are diploid because you have both sets. But this is only one pair. You actually have many pairs of chromosomes. You have 23 for a total of 46. All of these chromosomes are completely identical to each other in terms of what genes are there, but they might have different alleles. There is one exception, and that is the sex chromosomes, which we will talk about today. So sex chromosomes, basically if you're a guy, you will get an X and a Y, which is what this thing is. And if you're a female, you will get two X's. And so from this we can see genotypes. So for example, the genotype here is gene A, and here it is gene B, and then down here, this would be the first one that we have, or sorry, second one, that is heterozygous. That's how we get genotypes. And so again, this really is the same thing as, like, say this. It's the same thing. It's just has been through S phase. Still two chromosomes. It's just these have sister chromatids. That leads us to today's topic, the chromosome theory of inheritance. And so again, we've been talking about alleles being inherited from each parent, right? If you have this, you have one allele from your mom and one from your dad. But those are actually found on chromosomes. And so that has some implications. If we look at these chromosome pairs here, let's say this is from your mom, and it splits into two eggs. Well, that means that you're going to get some package deals. So perhaps you will always get recessive A and recessive B together, or dominant A and dominant B together. And so, for example, if I have if the mom here is, well, she's heterozygous. So if she's got dark hair and dark colored eyes, the two different eggs she can make would be, let's say capital A is dark hair, and capital B is dark eyes. That's easy, just like her. But let's say that the recessive form is orange hair with blue eyes. That would be that. And so you would either get this combination or this combination, and that's what we call linked. When alleles are found on the same chromosome, you typically will inherit them together, like a package deal. So in this example, blue eyes is a package deal with the allele for orange hair. You will always get them together from this particular mother. Now, that's not to say that you will definitely be this. You have to look at what the father is, too, and get a mix from there and do a Punnett square. But this is the genes that the mother can contribute. It's either package 1 or package 2. So a package deal is sort of like if you get an Xbox and they give you a free game with it, like the new Assassin's Creed Valhalla, you would get that with it. That's a package deal. Some traits are inherited together. Like, for example, it's very rare to see somebody who's got a dark complexion, like African American or Latino, have blue eyes, right? That's very rare. Usually it's just white guys, right? Or girls. So there are exceptions, though. We do see that sometimes. So, for example, look at this kid. That's pretty rare. She's got a dark complexion. I think she must be like Indian or Latina. I don't know. But she's got some blue eyes and she's got a dark complexion. That does happen, although very rare. And so let's talk about that. Let's say that her parents had gene A here for brown skin and gene here for brown eyes. But then another parent had this and this, right? So let's oh, shoot. There it is. Sorry. Here we go. Here's where all the magic happens. If you remember from the last chapter, meiosis comes in 2 phases. There is this phase, which is meiosis 1, in which we have prophase, metaphase, anaphase, telophase, and we just put 1s next to them, right? Because it's meiosis 1. Whereas in mitosis, we don't put 1s or anything next to it. And then in meiosis 2, we then split the sisters. So up here, we're splitting homologs. Down here, we're splitting sister chromatids. So the magic is all happening right here during prophase. And again, this is meiosis, so we have to designate what phase. Prophase 1. All the magic is happening right there. So let's take a look at what that magic is. Actually, we can just go here. This is what's happening. If you got this chromosome from your father and this one from your mother, or sorry, reverse. This one from your mother, the red one, and this one from your father, the green one. That could create some interesting mixes. And so let's say that, for example, the father had the dark-colored eyes and the dark-colored hair, but the mother had the light-colored eyes and the light-colored hair, right? So when we do crossing over, we form what's called a tetrad during prophase 1 of meiosis, and that could mix things up. So we could, for example, get the lowercase a with a capital B, and then we could get the uppercase A with a lowercase b. And so, of course, if you look at meiosis, there's 2 rounds of division. The first thing you divide up is this during meiosis. So that's meiosis 1. And then during meiosis 2, we will divide up these. And so you, being the child of these parents, of this particular parent, you will either get this chromosome or chromatid, this chromatid, this chromatid, or this chromatid. And so if you got, for example, this chromatid, now instead of having those linked genes, we have now crossed them over. We have unlinked them. And so you could, for example, if we were to talk about, I think we said capital A was dark hair and then lowercase a was orange hair. You could get an orange-haired child, and now they have the capital B, so they have dark eyes. And so they are no longer linked. So you can unlink genes with this crossing over thing, and that creates a huge diversity in potential. This reiterates Mendel's principle of, what was it? Independent assortment, yes. And so we've just done a 180 here, sorry, a 360. We did a 180 here. We basically said, okay, genes are linked. That means that Mendel was wrong. There is no independent assortment. Some genes have to be inherited together. But then we said, oh, wait, but crossing over can happen. So actually, Mendel is correct. You can get any combination of traits that exist. So there's a truth halfway in between this, right? And that is crossing over can happen, but it's unlikely for some genes. So if we look at, for example, a chromosome like this, and we have gene A here and gene B here, and then we have gene C here. And then we have another parent who has the opposite profile of genes. Crossing over is much more likely to happen between gene A and C than it is between gene A and B. And that is because there's more places between gene A and C where those chromosomes can touch and make the switch. I like to make an analogy of a ruler. Let's say we have a ruler, and instead of a chromosome, and instead of genes, we'll use inches. So 1, 2, 3, so we'll say 1 and 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. Between gene A and C is like saying between inch 1 and 11. So if I were to be blindfolded and I were to take a axe and bring it down on this ruler, and I'm blindfolded, I can't see anything, would I be more likely to separate 1 and 2 or 1 and 11? In a similar way, when two chromosomes cross, are you more likely to separate gene A and B or gene A and C? The answer, of course, is you're much more likely to separate alleles in crossing over the farther they are away from each other. And so some genes are really hard to separate. Some genes are just nearly impossible to. I'm trying to think of an example in humans. So, for example, if you have I don't know. I'm having a hard time imagining. If you're really tall, you probably have high bone density, because they're probably really close together. That's just a guess. All right, so that's linked genes, and now we're going to go over to sex-linked genes. So sex-linked genes are found on a particular chromosome pair. So let's go back and look at karyotype. Again, this is a karyotype, so I'll spell that out for you so you can put it in notes. I do hope that you guys watch these lectures with a notebook. If I never said that before, it's because I thought it was obvious. You should be writing stuff down. So a karyotype is a picture of chromosomes. And I like this because you can tell a lot by it. First of all, you can tell the gender. Can you guess what the gender is of this individual? You're going to look here. So with a female, you have an XX chromosome, and with a male, you have XY. If you look at that Y chromosome, something funky going on, and we can see it better here. This Y chromosome is actually missing a lot of genes. So in fact, males are haploid for X chromosome genes. Here's what I mean. If there's a particular loci, like let's say this is the loci, and again these are sisters, so they're the same exact gene and allele. So let's say there's gene A here. Would also be gene A here. But this chromosome here does not even have the gene. It's missing information, right? You can see how small it is. Let me draw it, actually. So here's the X chromosome with its two sisters. And then we'll draw the Y chromosome. There's no gene A. And so effectively, this is haploid. Males are haploid. They're missing a second set. And so instead of drawing the chromosomes every time, what we just do is we just write an X for X chromosome and we put a superscript on it, just like we do with codominant genes. And then for the Y chromosome, we just write Y, which just means there is no gene present at all. And so that means that men are haploid for any gene that is found on the X chromosome, or at least most of them, right? There is some stuff on this Y chromosome, but it's usually found here too. All right, so what are the implications of this? Well, first of all, let's talk about who determines your gender. Does your mom determine your gender, or does your father? Well, to answer that, look at this Punnett square, right? Your mom is always going to give you the same thing, an X chromosome. Your father can only give you one. And actually, this Punnett square is a little bit redundant. We don't actually even need this row, right? Because again, a Punnett square just shows probability. There's no point in doubling a probability. That's like saying a 1-to-1 ratio is the same as a 2-to-2 ratio. That's the same thing, right? So why add this extra column, right? The mom can only possibly give you this chromosome. The father, however, can give you two. And so that means there's a 50-50 shot of being a male or a female, which should make sense. So who is determining your gender? Well, the mom can only give you one type of chromosome, so she's not determining anything. There's no choice there. The mom is definitely giving you an X chromosome. But the father gives you one or the other. So it's actually the father that determines your gender, not the mother. So this has some implications when it comes to certain diseases, like recessive diseases especially. We all know that there's 3 types of genotypes. With this genotype, the homozygous dominant, you have the dominant form. You also have the dominant form with the heterozygote. It's only this form that gives you the recessive. But what if there was no second gene? If you're a man, and let's say that there is a gene for color blindness, and we'll use B to represent blindness. So we'll say that you have a recessive gene, right? Well, there's no second chance, right? If you're a female and you have a recessive gene, you have another chromosome where you might stand a chance of being normal, because this is normal. You would need 2 copies to be blind, to be colorblind. So females have another shot. They are diploid. They have 2 chances to get the right gene. Males only have 1 chance. And so let's look at an example. Let's say that a father is perfectly fine. He is not colorblind at all. He got the good gene. But the mom, actually the mom is also not colorblind because she has 1 good copy. So theoretically, I mean, you would think that none of the children could be colorblind. None of the parents are, right? Well, if we start putting this together, so far so good. The daughters are all okay. Now let's look at the sons. This son is okay, but this son is not. This son is colorblind. And you see it's because he didn't have another chromosome there. He could not make a heterozygote. He is diploid for that colorblind gene. So that means men are more likely to get certain recessive sex-linked traits such as colorblindness, hemophilia, which basically means you don't scab up, you bleed a lot. Muscular dystrophy, which means you can't repair your muscles after they are damaged, like after you exercise. Your muscles are supposed to repair themselves, but if you have muscular dystrophy, you cannot. And other recessive traits on those X chromosomes. Who gave the son colorblindness, by the way? Whose fault is it when a son has a recessive disease? That's right, it came from the mother. So it's your mom's fault. If you are a son and you have color blindness, it's your mom's fault. It can't be your dad's fault. Because your dad, the only chromosome he gave you, if you are a guy, the chromosome your dad gave you was a Y, and that doesn't have anything on it. So it's not your dad's fault, it's your mom's. Alright, I think I'll wait and talk about mutations when I talk about DNA. So that'll do for today.